But we're going to move into our second session now. And here we're going to look at research dimensions of serosurveillance in the context of pandemic preparedness. So we have Dr. Jen Gommerman, who was with us yesterday, but some of you may not have been with us. So just to say that Jen is the professor and chair of the Department of Immunology at the University of Toronto. She works on adaptive immunity, autoimmunity, gut immunology, and multiple sclerosis. She's a Canada Research Chair in tissue-specific immunity, and she's been a major leader during the COVID-19 pandemic on issues of immunity, and also working with COVARNet, studying mucosal immune response to infection and vaccination. So Jen, I'm going to pass it over to you and the panel. Thank you.
Can you hear me? Okay. So I'm going to start by introducing our first speaker, Dr. Ligia Pinto, who, if you did not see her session yesterday, is the director of the Vaccine, Immunity, and Cancer Directorate at Frederick National Laboratory for Cancer Research in the U.S. This includes the HPV and COVID-19 serology laboratories, as well as the Cancer Immunoprevention Laboratory. Dr. Pinto also leads the National Cancer Institute's Serological Sciences Network for COVID-19, that's SeroNet, efforts at the FNLCR. Welcome again.
Thank you so much. It's really a pleasure being here today and attending the meeting. I'm really enjoying the meeting, and I'm looking forward for collaborations with IMANET. So to tell you a little bit about the Frederick National Lab is a federally funded research and development center solely dedicated to biomedical research, huge focus in the area of AIDS, cancer, and also diseases associated with pandemic agents. The HPV serology under my department has played a very important role in standardization of HPV serology. As I mentioned yesterday, we initiated a standardization initiative global across the world. And that has resulted really in very important public health actions with using serology as endpoints for vaccine effectiveness and really using it for novel vaccine recommendations and as well as new vaccine approvals at low-income countries. So the reality is that when the pandemic came in 2020, in March 2020, our lab, because of all this work on serology for HPV and all the partnerships we're talking about here, how important collaboration is and having a network was much smaller. We started in 2016 building this HPV serology network, but was critical for us to quickly pivot to COVID-19 in a very fast manner. So this shows you, again, the example of how can you translate knowledge from one infection to the other, from one situation to the other. So we were asked by the National Cancer Institute to help with COVID-19 serology. This is end of March. At the same time, FDA, so we are actually a WHO reference center for HPV serology, and we became an FDA testing laboratory. So the FDA asked us to help evaluate the quality of the emerging commercial serology tests that were submitted to FDA. As we were discussing at the beginning, the market got inundated with all these serology tests, some of them with very poor quality. And people were assuming they were fine because they were coming from sometimes reliable commercial sources. So what was really important was to establish again a network. So it was a very large collaborative effort. So this was an evaluation program, independent evaluation program that was established between FDA and different agencies of the government. So NIAID, CDC, BARDA, also NCI designated cancer centers. And really the NCI directors at that moment, Dr. Sharpless and Lowy, had a very important role in the establishment of all this work. They were extremely excited, as you were saying, zero sleep for all of us for about a year. The colleagues at FDA, CDC, several agencies were working day and night, again, to test these devices, to build those validation panels that were needed and those standards. And you can see all the device evaluation is actually open to the public, both at the FDA site and also the CDC site. So you can see the performance characteristics of all these devices. And then that work really led to congressional funding for serology research. So the funding really allowed NCI to support a wide range of activities, including the establishment of a network. We all felt, and the network was really initiated as a model of the HPV serology standardization network. We all felt we couldn't do this alone. 1 + 1, as I say to anyone, is not 2, but it's actually maybe 11. And if you can, you can actually look at the details of all our work on a recent publication in Lancet Microbe. So the goals were, again, to increase that national capacity for serology antibody testing and advance our understanding to this new infection that we had no idea about. So it was designed to be a very interactive network, very fast-paced. We were meeting several times a day, with different groups and any time of the day, really. Sharing of data and resources was very important immediately, and then open access publications. So in terms of the components, there was 25 different organizations involved in this network, mostly academic groups. So about 48 different institutions, because there was the grants and the subcontracts to each of these organizations, but then there was collaborators as well. So the goal was, again, to develop and standardize novel serological assays with high quality, deploy them broadly to the community, understand the innate humoral and cellular immune responses to infection and vaccination, because that's critical, a parallel exercise here. And then also there was funding to a number of epidemiological groups that were going to evaluate factors and determinants of the immune response. There was the funding went to a number of grantees, 8 centers of excellence, 13 UO1s, 4 serology testing capacity centers through contracts, and then our laboratory, the Frederick National Serology Lab, as well as our data coordinating center. And then this shows you the map of the distribution. So really, grants were distributed in a wide geographical area, as well as the subcontracts, again, to have representation of the country and the populations of interest. And then we had a hub model in which we had the data center and the lab, very important in establishing really the infrastructure of good science, that is standardization, very important to really develop validation panels and provide SOPs and best practices to all these groups. So in terms of key contributions, these are just some of the contributions. Again, we had a key role in establishing this independent evaluation program with FDA. That was critical to really evaluate the tests that were submitted for emergency use approval. We tested more than 120 commercial tests, and only about 1/4 of those fit the performance criteria set by FDA. Some were actually revoked. The UA was revoked and were taken out of the market. We produced and distributed the US serology standard, working very much in collaboration with our previous partners in the HPV Serology Initiative, WHO, and NIPSC. We implemented and validated the assays, so that was very important, included also multiplex assays, as well as other research assays. And we investigated them using these tools and validated tests. We looked at immune response to the licensed vaccines and to infection. And then, as I was mentioning, the coordinating center was instrumental for the preparation of all the meetings, managing the subcontracts and the data curation for all the different publications. I have to say that Seronet had more than 400 publications. They are all in import. The data is shared in import. I have to say, I was looking at the number of publications with seroprevalence. This is not a serosurveillance, national serosurveillance network. Is more a research network, but there was a lot of seroepidemiology. So there was about 20% of those publications were around seroepidemiology studies. And we established also the clinical and translational task force. What I want to point out is that there was that publication in New England from our collaborators at FDA that really emphasized the importance of creating these networks for independent evaluation, because not everything that is commercial and that it may show good performance on their hands is going to show good performance in independent evaluation. So that's a very critical component. So the capacity building centers, just to tell you a little bit more about it. So it was Feinstein Institute, Northwell, Arizona State University, Mount Sinai, and University of Minnesota, and their goal was to develop standardized serological tests, high quality. This was really very important. Provide national capacity for serology testing, and they have been doing that. And I have to say that Mount Sinai and Northwell are still doing a lot of serological testing in a daily basis. And then critical, and this is relevant to Imanet, was really acquire large volumes of serum for development of serology standards and validation panels. You need a lot to create these validation panels that can be utilized, the same validation panel, which is about 120 samples, with the requirements set by FDA to analyze now 120 different devices. And then they were also responsible for conducting serosurveillance studies and technology development. Some of them developed increased and enhanced assays to look at mucosal immunity, neutralizing assays. So there was a number of really inventions that occurred at these centers as well. So I think one very important aspect, and I am a big advocate for longitudinal studies, is critical to understand immunity over time because it's very dynamic. So that's very important. It's very costly, but really you actually understand so much about the infection and the vaccination. And so the CBCs, the capacity building centers, in addition to collection of large volumes of sample for the standards, they also did longitudinal studies in different areas and different populations. So being sponsored by the National Cancer Institute, we are interested in cancer patients as well as immunocompromised patients. So in addition to healthy, there was autoimmune cohorts, cancer cohorts, HIV, transplant recipients, all using the same sample processing SOP and also the same data elements. I have to say that there were 4 working groups within Seronet. One was serology assays, the other one was cellular immunity assays and animal models, the other one was data. There was a data group, and there was also an epidemiology group as well. And they met weekly, really, during really the first 2 to 3 years of the network. And in terms of collection points, time points, they were also harmonized. So we want really do collections in an intensive manner to better understand the immunity. And we were collecting not only serum, but also high quality PBMCs collected within the same day. So I just want to tell you how really the timeline was incredible. Because everybody was very aligned, very important. You had the right expertise, the alignment, you had previous agreements in place, templates that we could use, the connections as we were discussing today. And so, for instance, the materials were received in our lab around end of March. Within 1 week, with our plug-and-play technology, we had the ELISAs working for SARS-CoV-2, and then 2 weeks after that, we had the validation panels ready to start testing the assays. At the same time, the grants were being really, the RFAs and RFPs were being published, and then it was a remarkable timeline to get all the grants and contracts awarded a few months after the RFP, and then the serology standard became available. We have distributed to over 200 labs, and also the assay validation panels. And I have to say that if you go to our website, anyone in the community can request the serology standard as well as the validation panels. For the longitudinal samples, we are, and I'm trying to really make them available to the outside. Right now is available to the SeroNet investigators. Important was this task force, and the co-chair is my colleague, Dr. Cardon Cardo from Mount Sinai. We still meet 2 to 3 times a month. And we actually, in this group, I think what is important is bring everyone with the right expertise and really a multidisciplinary group of individuals like you have it here to really discuss important topics. And so we continue doing this. We publish them, and you can go to the website, and we have representatives from CDC attending, WHO, not just the academic centers or NIH, and you know, some international groups as well. And I think what I want to emphasize here is that we have very specific goals. We wanted to implement its standardization. We were successful with that. We wanted also, and I mentioned that in one of the sessions, how important it is to actually evaluate the landscape of the assays and then really do an assay comparison study where you can evaluate what is the best assay and technology for depending on your use. One size does not fit all. So we actually did that collaborative study in a very rapid manner with 27 different assays and 17 different laboratories. And that has been published and was following really the example set by WHO on standardization and again, building that capacity for testing, again, with the highest quality and fit for purpose. And then again, how important it is to have regulators and public health experts and also associations. Really, we involve IDSA in some of the meetings, so we can actually think about development of guidelines and recommendations. And I have to say that we have underutilized serology still as of today. And I think there is a huge possibility with IMANET to really leverage the power of serology to the next level that we want to see. And so examples of those collaborations and that task force was really, we built a real world data infrastructure, which is led by Lynn Penberty at the National Cancer Institute and Jim Crawford at the Northwell, one of the capacity building centers. Actually, there is a call for applications right now until March to look and do analysis on about 5 million patients, cancer as well as healthy individuals, in which we actually, for the first time, we are linking serology data from Quest and some other, Northwell and some other testing centers with the electronic medical records, which I'm really excited for that analysis. And then we also, another outcome was really building that center of excellence, which is really SeroNet Phase 2, for that development, lessons learned, the development of agile sample acquisition, as well as very high quality serology testing and other immunological testing. So one of our goals within this Center for Pandemic Preparedness and also serology development, we want to standardize T cells. What I saw within SeroNet is that we have hundreds of publications on T cells, huge knowledge, but there is not one FDA approved test that came out of that work. And I want to see that translation to public health. We know T cells correlate with severity of disease. So why not using that for recommendations in the future? So that's one of the goals we have, is bring T cells assays to the level of the serology standardization. It's going to be challenging, but we have that goal in mind. Again, we have been talking about these gaps and the solutions, that strong network of expertise, capability too, because people sometimes are extremely excited and they have the expertise, they don't have the bandwidth, they don't have the capacity. So it's very important to identify the groups that actually can do it, what we need to be done. And then in terms of, we have been discussing where to apply ImmuNet, and in terms of the long terms that we see for all this infrastructure that was built, this knowledge, these networks that are so important, is really applying it not only to COVID-19, emergency preparedness. Again, we have these plug and play technologies that you have here too, and you are intending to apply. And also for cancer. And so we are very interested in really applying it to the cancer space in the context of cancer vaccine development. And then it takes a village, as you all know. But I want to thank the leadership from the National Cancer Institute, as well as all the colleagues from FDA, amazing work, BARDA, CDC, NIBSC, NIAID, and my team at the Frederick National Lab. Thank you so much.
That was awesome. Thanks, Dr. Pinto. Our next speaker is joining us virtually. Dr. Melissa Coughlin is a lead biologist at the Centers for Disease Control and Prevention in Atlanta, Georgia, with over 15 years of experience in microbiology and immunology. She's currently the head of the Human Immune Response Team in the Coronavirus and Other Respiratory Viruses Laboratory Branch. Currently, her team specializes in the humoral and cellular immune response to non-influenza respiratory viruses with a focus on SARS-CoV-2 and RSV. In previous roles at the CDC, she studied the immune response to measles virus and implemented assays to study measles and rubella serosurveillance globally. Welcome virtually to Montreal, and looking forward to your talk.
Thanks. Just want to make sure that you can see the slides okay. Sorry, if you could confirm that you can hear me and see the slides.
Yes, we can hear and see the slides.
Thank you. Thanks for inviting me today to give a short talk about the seroprevalence studies that we've been doing at CDC to look at seroprevalence in the United States for SARS-CoV-2. So serology has been an important part of surveillance for SARS-CoV-2, and as the pandemic has evolved, the public health surveillance that was case-based has changed, and so currently the ongoing public health surveillance includes case-based surveillance on deaths and hospitalization admissions for SARS-CoV-2. And therefore, it's only the top part of this pyramid that you see on the left. And so unreported cases, including those that are mildly symptomatic, are not captured currently through our public health case-based surveillance. And this is where serosurveillance can be used to help estimate the total number of infections. So we have a number of different avenues that were pursued to look at serosurveillance at CDC. I'm only going to talk about the top two, which is the National surveillance, as well as leveraging some of our existing surveillance networks. There was also a number of cohort studies that were done looking at specific populations, but those I'm not going to go into today. So one of the national level serosurveillance studies that was conducted at CDC was based on commercial laboratory. So these were residual blood specimens from commercial labs, and this included serosurveillance of individuals of all ages. It was shifted in 2022 to focus primarily on the pediatric population. So I'm showing a little bit of data from that evaluation, and so this is national seroprevalence that was tracked using these residual specimens between August of 2020 to February of 2022. This, what's shown here on the right, is the last collection date for the anti-N antibodies that were in February of 2022. And so this would be in February of 2022, the national level of N reactivity was about 58% across the United States. And you can see the range based on the jurisdictions with the lowest and the highest states. This data is all publicly available on our COVID data tracker. As I mentioned in March, this study shifted to focus on primarily the pediatric populations in March of 2022. They continued to use a qualitative anti-N assay as the results from the previous slide were generated from, but they also added in quantitative anti-S antibody reactivity. And so what you can see here is the breakdown of the infection-induced as well as combined seroprevalence in children from 6 months to 17 years of age, broken down in the different age bands, with the highest seroprevalence noted in those 12 to 17-year-olds. Another national level seroprevalence study that was conducted and continues to is continuing to this day was based on a blood donor seroprevalence. And so this used testing of blood donors from 2 large US donor organizations. So it included everybody age 16 years of age and older that donated blood. These blood donations were evaluated quarterly. And then within each group, there was a shift in 2021, in the end of 2021, to look at longitudinal samples from the most frequent donors. And I'll talk a little bit about that. So this is the combined seroprevalence for the US population from October to December of 2022. This includes both anti-S and anti-N reactivity. And again, you can see that the seroprevalence in the United States is extremely high by the end of Q4 of 2022, with the national combined seroprevalence of being 96.7%, and the infection-induced seroprevalence from the anti-N reactivity of 77.5% during this time frame. And again, you can see the ranges for the jurisdictional seroprevalence for the national level is quite homogeneous across the states. This too is also publicly available data on our COVID data tracker. As I mentioned, in the end of 2021, there was a shift for the evaluation of samples from the blood donors, and this shifted to a longitudinal US blood donor cohort. So there was 142,000 frequent donors selected. They received quarterly surveys that evaluated their vaccination and infection status. And then based on that information and the antibody reactivity, they were grouped into four groups. So those that had no infection or vaccination, those that had vaccine-only induced antibody, those that had infection-only induced antibody, and those that would be considered hybrid immunity that had both infection and vaccine-induced antibody reactivity. And so the primary outcomes of this evaluation was to look at the risk of infection and reinfection. There's also a component of this, which I won't really go into today, where we looked at or are looking at the humoral immunity in terms of the antibody waning, the stability, and the boosting that occurs with reinfections. And then part of this also is being applied to vaccine effectiveness and understanding correlates of protection. So this shows some of the data from that longitudinal cohort from Q2 2021 through Q4 2022, and you can see how the different groupings have shifted over that course in this river plot, and so how individuals have shifted from having no infection or vaccination. You can see in the orange how that's a very low proportion by the end of Q4 2022, and whereas those that had infection induced and both infection and vaccine induced antibodies have grown by Q4 2022. So as I mentioned, that this shifted to a quantitative evaluation of the spike antibodies. And so this is the quantitative titers of anti-spike antibodies in binding antibody units. So this is quantitated against the WHO NIBSC standard, international standard. And so the median spike antibody levels increased from Q1 2022 through Q4 2022 in the overall longitudinal cohort. And what we see, very similar to other data that's been published, is that those individuals that were infected only and had no vaccination tended to have lower spike antibody, whereas those that had were vaccinated or were vaccinated and infected had higher antibody, with slightly higher median antibody titers from those that had hybrid immunity. So just to touch a little bit on one of the other aspects of serosurveillance at CDC, during the pandemic, existing surveillance networks were leveraged to also include SARS-CoV-2, and those are continuing to include SARS-CoV-2 in their evaluation. So these include the US Flu VE network, as well as the IV network. And so both of these networks have a couple of different components. They look at infected individuals, each coming from a slightly different perspective. And then the other component of both of these networks includes looking at healthy vaccinated volunteers. So just a picture of the US Flu VE network, just to show the spread across the sites in the United States. This is Duke is the coordinating center. And similarly, the IV network, which is a case control based design. And the main difference here between Flu VE and IV is the infection based groups. And so IV looks at severe infection. These are hospitalized patients, whereas the Flu VE network looks at outpatient settings. And so those are people with more mild disease seeking care. In both of these networks, we collect a number of samples that includes samples for molecular testing, but we also collect acute blood within 5 days of symptom onset, so less than 5 days of symptom onset, and then we collect convalescent samples. So in the Flu VE network, for the component focused on infected individuals, that blood collection is 3 to 6 weeks for convalescent. And then in the IV network, in addition to that 3 to 6 week time point, we are also collecting 90 days. In these samples for blood, we look at the serology, but we are also looking at the cellular immunity. So based in serology, we look at binding antibodies as well as neutralization. And then we are also collecting PBMCs from these individuals that are willing to provide them. And those samples go on to look at the T cell and the B cell responses. I'm just showing a little bit of a snapshot of some of the data that we've gathered. This is from the IV post-vaccination cohort. So I mentioned that both of these also include healthy volunteers that have been vaccinated. So specimens are collected pre-vaccination at the time before they receive their vaccine or updated booster, and then 3 to 6 weeks post vaccination. And so what you can see here is that binding antibodies increase from the pre to post. This data is from the monovalent booster dose for both the ancestral strain as well as the variant spike proteins. This was all evaluated based on MSD testing in this study. And then on the very far right, as expected and shown by many others, is that we see about a 3.5 to about a 5.5-fold reduction in antibody reactivity compared to the ancestral or origin strain when looking at other Omicron spikes post monovalent booster dose. And that we can see some key mutations where we actually can detect differences in the binding antibodies as shown for the R346K mutation shown here. So just to summarize a little bit, there's high seroprevalence within the US, and that the concentration of spike protein or spike antibody reactivity in vaccinated and individuals with hybrid immunity is higher than those that were infected alone. The quantitative antibody measurements we feel is becoming increasingly important, and so we see that the spike antibody concentrations are increasing in the population over time, and that this also collecting the quantitative measurements helps to contribute to some of the correlates of protection analysis that we're doing. We have with the high seroprevalence that we are seeing now with SARS-CoV-2, cross-sectional data becomes a little bit more limited and difficult to interpret. And so many of these studies have moved to collecting multiple specimens. The other aspect that we're looking at is the measurement of boosting of the antibody response to detect reinfections, which is important to help provide more information about that base in that original pyramid of the number of infections occurring and the disease burden, as well as to estimate vaccine effectiveness. So just to acknowledge, there's a couple of acknowledgement slides here. Many of those that contribute in our Corvid, particularly Dr. Jefferson Jones, Dr. Suri, and Dr. Sumner, who contributed to some of the slides that I used to show you today, as well as participants in our IV and flu VE network, the folks at CDC that conducted the commercial laboratory surveillance studies, and then a very large group for the blood donor surveillance. And with that, I can take any questions at the end of our panel discussion. Thank you.
Thanks, Dr. Coughlin. So we're now going to move into the panel part. I want to reintroduce to you Dr. Allison Spalding, who's sitting with us here and has joined the panel, who spoke yesterday. She's an epidemiologist with expertise in infectious disease, pandemic preparedness, and antimicrobial resistance, and she is a senior advisor and team lead at NIAID and presently leading PREMIS, the Pandemic Response Repository through Microbial and Immune Surveillance and Epidemiology program. So with that, we are going to start with a few questions for the panel, and then we'll open it up to the rest of you. Dr. Pinto, I actually wanted to start with a question around the public. And so you mentioned at the beginning of your talk about these antibody assays that came out that kind of sucked. And, and how, a lot of, there were some very big studies that used these assays and came to the conclusion early on in the pandemic that once you got infected, your antibodies would only last for a few weeks and then you would be completely vulnerable once again. And when you looked into these publications, it was clear that they were using suboptimal assays. What is our responsibility and role, and how do we communicate with the public when these types of results come out? And why were these assays so sucky?
Yes, there was really
Please speak into the
inundation.
microphone.
Okay. Can you hear me? Yeah. Yeah, there was an inundation of commercial tests coming from all sources of groups. So that never worked in infectious diseases or diagnostics. And at the beginning, they could come to the commercial space without the emergency use authorization. As soon as they had, specifications followed, demonstration of certain specifications. But then it came the emergency use authorization, which really helped, because then they could only come to the commercial space if they received emergency use authorization, and then that data was available. Communication is very complex, as we know. Within the Serological Sciences Network, there was a group of individuals, and there are still dealing with communication issues. But again, I was interviewed by a few magazines and a few places about this. We wrote a few things about it. We wrote papers. But I think that, communication is critical, but over-communication can also be a danger, because if it's too premature, then you get those type of, issues coming. So I think with COVID, there was a lot of premature communication, inappropriate, that really caused a lot of, issues. And again, I think this type of networks is very important to have also patient advocates in the groups. So actually, Seronet, these patient advocate groups are included in our meetings, and they provide feedback. And again, there was a component of communication, but I think we could have done globally a much better job.
Yeah, and the media loves a bad news story, right? Mm-hmm. So, and we also saw, of course, the advent of preprints. I mean, they existed before the pandemic, but they became really a major form of communication between scientists. But I think the media used a lot of these preprints as well. Do you think there's a role for these types of networks in reviewing preprint material to provide an expert opinion on this for the media?
Yes, definitely. I think that, again, we created the Sero News, where we actually compile scientific information that is current. But again, that's us communicating what we are finding, our opinions, but people can go to the other, publications, and that's free, It's very difficult. It's going to be very difficult, and I'm not a communication specialist, but I think each network should definitely have and involve expertise in communications, and they have an important role in publishing, and also, in our case, work that we actually did with FDA revoked some of the testing, and that information is also very important to be placed in the public. So, yeah.
So, Dr. Coughlin, I was curious about how you looked at sero data for people who were infected versus people who were infected and vaccinated, so hybrid versus infection alone. How did you discriminate between those two groups? Is that where quantity of antibody is coming into play here? I was a little curious about that.
Yes, sure. Thanks for the
Can you
question.
speak up, please?
Yeah, can you hear me?
No, we can't hear you. Try again.
Can you hear me now?
Yes.
Okay. So for the difference between vaccine-induced and infection-induced evaluations that were done was all based on anti-N reactivity. And so in the US, all the vaccines that are approved are spike-only based vaccines.
Yeah.
And so
I was more curious about infection versus hybrid.
So those were based on the survey data. So in that, yes, so in that cohort, there was quarterly surveys that were performed, and those individuals answered questions about their vaccine doses as well as any infection history that they had, and then the anti-spike and the anti-N reactivity was evaluated in every.
Okay, that clears it up for me. Thanks. Do we have any questions from the audience while I grab another question? think we should move to general questions. While we're waiting, I loved the comment about cell-mediated immunity and how this could potentially be used to translate to the public health space, because we know that cell-mediated immunity we learned a lot about who was having the most severe concerning infections. How do we do that? I mean, this is a plug for, I think, large-scale biobanking with large volume leukapheresis samples. But I know this is a serosurveillance conference, but I think these two are entwined. So just curious about your thoughts on that.
I completely agree. I think that, again, we can't have silos anywhere. Immune system is not a silo. So we can't just say, oh, we are only going to study. I mean, we have to focus and we have to come up with priorities. And I was a T-cell immunologist in my prior life, and then I became a serologist about 20 years ago. So I love both, and I think it's so important to understand both. And I think we need to leverage them for public health potential, because as we saw with Seronet, amazing papers and amazing panels for T-cell characterization, but the variability is very high. And when you talk to regulators about it, they say, yes, submit a package with the right data, with the right claim. You want to say that, you know, this serves for this purpose and submit the packages of data. But then when we see everything that's required, we are not ready with T-cells. So I think this is where the communication, if we want to translate to public health, we need to involve regulators and public health experts, of course, decision makers. But we need to make sure that we actually, from the onset, we prepare the data to be ready to fit with those regulatory and public health requirements. So we are not there with the T-cells. Again, that standardization, that validation according to GCLP is going to be very important. It's not easy, but I think it's possible to get much more and then involve also commercial groups at the same time that can actually bring this to the commercial space.
And I think that brings me to the design of Seronet. You mentioned these 4 pillars: EPI, CMI, so cell-mediated, animal modeling, and serosurveillance. And those 4 things really need to talk to each other. And then you had this global MTA, and I assume ethics as well, REB. Like, how did you do that? Because that's what we need to do here.
Yeah, definitely. So we had, again, we had MTAs done in 24 hours with organizations, commercial organizations. The reason for that is that we had a template agreed to use for the HPV, because we were providing, again, we are actually providing testing as well as capability and training groups globally for HPV. So we had all this ready to be able to provide standards to low-income countries and globally. So MTAs were done in about hours. And then there were Seronet agreements within each organization. So that really, having those templates ready was really important to an expedited actions. Yeah.
I think we need to advocate both to our intramural and extramural leadership that this is non-negotiable. We have to move forward. Angela.
Great, thanks so much for this panel. So just to pick up again on the CMI bit, so given that the intention is around surveillance, do you find that you hit up against limitations that allow you, like, because CMI is serology is expensive. CMI blows the barn door off. So do you find that, both the mandate to serve the surveillance and the academic and research interests to go deep, right? So as soon as you touch a T cell, it is imperative that you go deep. And so that starts to take you away from the surveillance piece, and then the deep mechanistic hats come on, as you know, as a T cell scientist. So do you find that you hit up against these limitations, which speaks to, the need for parallel efforts in different sectors, like the extramural component, right, where you have the research entities and the surveillance entities, finding that overlap so that those limitations can be overtaken, but they don't have to necessarily fall within the purview or the responsibility of the surveillors, if that makes sense.
Yeah, I completely agree. This needs to be really segments, so that will be blocks that are moving in parallel. and with what I think is very important is set goals from the beginning very clear. Again, solutions. What's the goal? What's the solution? Select 4 or 5 at the beginning, and then make sure that people are working in a coordinated, it needs coordination, it needs communication. A lot of colleagues in academia, again, I work with, regulatory bodies and public health, advisors, and the reality is that a lot in academia, and when I was in academia too, you tend to think, oh, it's so difficult, it's so this, oh, why do they require this and that? So I think, but when you actually talk to the individuals and you understand the process and transparent communication in terms of workflows, a lot of things you just don't know how to do it because you have to talk to about 20 different individuals to figure out a workflow that if that existed, that SOP, that workflow, it will take just maybe a week instead of 10 years. So I think establishment of workflows with involvement of, again, all the right expertises is going to be very important, and in moving things in parallel and bringing in industry to the table, because again, we have technologies that we heard yesterday from Dr. Mina that can we apply, and what's the next generation of these cell assays? Maybe we can come up with some panels that are going to be much cheaper and much easier to implement in, let's say, low-income countries. So, but we need to really, establish some of those goals in mind, and make sure that there is that communication.
Dr. Spalding, do you want to add to that?
Yeah, I'll just add to that, from the translational side, as I spoke about yesterday, obviously for our work, we need PBMCs. So as I think about that in the HemaNet context, because I've been trying to spend a lot of time thinking about that, and hopefully I can continue to be involved in helping to form it, because I so fundamentally believe this is important. I think, there's the cell component that we've talked about. How do you sell this to people to fund and support? And I think the research and the translational piece has to be a part of that, but it doesn't necessarily need to be part of the phase 1 of the implementation. And so if you are to get HemaNet off the ground with existing samples, for example, if you had the possibility to then build in consents moving forward for future sample collections to say, can we reach out to you again for additional research questions we might have, additional sample collections we might want to do, then that'll allow that translational piece to happen a lot easier, I think, moving forward.
Dr. Coughlin, you want to add anything?
Sure, I agree that I think the nature of the work makes it more difficult. So we do include cellular immunity in our different platforms, primarily the network-based platforms that we're participating in, but those are always only a fraction of what we would collect for serology. And so I think that there's a limitation in the capacity of what you can actually run in the laboratory. And I agree that the variability exists much greater in the cellular aspect when from individual to individual. And so capturing that, I think, is going to remain a challenge. But I agree that looking at the cellular immune component is incredibly important to understanding the immune response, as well as to infection and to vaccination.
We're just going to have to science our way out of that one. Tim.
Yeah, so just to build on that, I think science is moving quickly, so we may be able to science our way out of there faster than we think. But first, thanks. Terrific to see this activity. You're now in sort of the late phases of monitoring. I'd be interested in what your sustainability plans are. We've seen in the context of SeroNet an very impressive group of laboratories. Is there a sustainability plan that's going to keep that going post-pandemic? Also, in the various networks that are being drawn on by CDC, IV, FluV, and also I saw very interesting collaboration across the 2 primary blood bank, blood donor services, right? National Blood Bank and Vitalant, or something like that. Yeah, Vitalant. Yeah, so is there a sense from your perspective that those things can be sustained so that they're accessible and available in the context of the next threat or other conditions that are already on the horizon?
Yeah, so the networks that we work with were existing already, and so those were leveraged for including SARS-CoV-2, and we are moving forward to actually include other respiratory viruses within some of those networks. At least Ivy right now is also looking at RSV. So we hope will continue to be part of sources of information that we can understand these infections and the burden of disease, as well as the immune response. In terms of the blood donor evaluation, there is a contract call, a BA call that is currently existing for continuing that kind of work. The work that was presented was through Vitalant and American Red Cross, but I can't speak to the next phase of that. But we are hoping to move to look at additional respiratory viruses in our platform. So like I said, with the networks and hopefully with other larger national level based surveillance, we could do this.
Dr. Spalding, do you want to add to that, or do you have to wait till after November?
I think it's March, actually. I think I think we have an upcoming continuing resolution. So our work, given that we're intramural, we're not as impacted with the sustainability, but our partners desperately are. So, we have our staff, we have our laboratory capacity, but it's just us and our team. And so any of our partners we work with are, the funding issue is definitely an issue and gonna, I think it, become more of an issue, and especially because, again, as I mentioned yesterday, we work in a lot of viruses that don't have necessarily huge portfolios of work or industry interests behind them.
Yes, sustainability is a big problem. And the funding, continued funding for, maintaining these infrastructures. So at the moment, there is funding, there's still funding going on, but we'll have to seek new funding for future activities. So definitely.
I just want to come. So maybe as a follow-up, we could do a little bit of economic analysis to look at how much money was put into these networks, because I think part of what we need to think about moving forward is what constitutes value for money, and it may be that your unit costs are very different from what CITFs were or others. And I think that might help us get a sense of making the case in the context of tight fiscal resources on where and how we might get that, the rationale for continuing some form of this type of networked activity.
I want to finish up by a rapid fire. I want you to say the first thing that comes to the top of your mind. Based on serosurveillance research conducted during COVID-19, what lessons have been learned that can be applied to future pandemic preparedness? Don't overthink it, just answer.
Again, more structured and coordinated activities is still needed. There is still a lot of silos, elimination of silos. And again, I think the other thing is trans-network activities will be important, because that way we can do more for less, for sure.
So coordinate, eliminate silos, eliminate overlaps.
All of that, plus I think if we can try and focus on some of the, I don't want to call them positives, but some of the things that came out of the pandemic that we can use for forward momentum. And I think the bringing into the public knowledge monoclonal antibodies and what they are, what they can do, I think we can leverage that moving forward.
Some good news stories. Yes. Dr. Coughlin.
Agree with everything that was said. I think really kind of a key point takeaway for me is that what's been built, let's try to sustain it or see how we can continue to have those platforms. I think having the platforms and the ability to get the samples was the most important part, and the largest hurdle, and tends to be a large expense. And so I think that part is getting the samples is important.
Convince the funders to not kill the baby. All right, thank you very much. I'd like you to join me in thanking our panelists and speakers for this session.